And our next speaker is Jia Ling, who's going to talk to us about enhancer specificity at the hunchback locus.
Hi, I'm from Steve's Small Lab, and today I'm going to show some of my work on enhancer promoting actions. So as we all know that enhancer mediates transcription by contacting the promoter of a gene. In the genome, one enhancer is always located among multiple promoters, and to ensure a correct spatial temporal expression pattern, the enhancer needs to recognize its target promoter and activate it specifically. So how do enhancers contact specific promoters? To investigate mechanisms controlling this process, we studied enhancer-promoter interactions at hunchback locus. The regulation of hunchback is complex. There are two promoters. The P1 promoter is thought to be expressed maternally and late zygotically, while the P2 promoter is expressed early at the anterior part of the embryo. And this pattern is driven by bicoid, which you are all familiar with. The bicoid binds to a proximal enhancer, which is next to the P2 promoter, and the proximal enhancer drives the expression of P2. The bicoid also binds to a distal enhancer, which is 5 kb upstream. You can see it is more closer to the P1 promoter. So it raises the question that whether the distal enhancer interact with the P2 promoter or P1 promoter. By generating simple reporters where the distal enhancer is placed before the next to a promoter, we found that the distal enhancer drives the expression of P2 as the proximal one does, but it counter-activated the P1 promoter. So does it suggest that in vivo the distal enhancer will skip over the P1 promoter and contact P2 promoter instead? We can't get an answer from these constructs because they are very different from the endogenous locus. So to answer the question, we built dual reporters mimicking the endogenous hunchback locus, including both bicoid-dependent enhancers and the two promoters. And in order to quantify the outputs from the two promoters, we introduced a single molecule fish approach. To do this, we split one whole length lacZ into two halves. Each half tag one promoter, and each half has a set of probes hybridized into it. And to get the number of transcripts, a small region at about 30% egg length was imaged at a certain stage where there is no active transcription. And the cytoplasmic dots were representing single transcripts were counted and scaled to a number per nucleus. And since we want to compare the difference between the levels from P1 and P2, we want to first make sure that the difference we see is not from the different capabilities of the two probe sets. So we tested the two probe sets on this whole length lacZ construct and confirmed that they identify the same number of transcripts. Okay, so with this experiment setting, we found that this reporter mimicking the endogenous locus can recapitulate the early hunchback pattern. Basically, the P2 promoter is highly expressed, but the P1 promoter is expressed at a very low level compared to P2. So this confirms that in vivo, the distal enhancer does skip over the P1 promoter but activate the P2 promoter. So why count the P1 promoter activated. It is because that P2 promoter is competing with it for enhancers. So we removed the P2 promoter in this context, and this reduces the lacZ, the C half lacZ expression, but it does not activate P1 promoter, suggesting that competition from P2 promoter is not the answer. But as I mentioned, and you can see that there is a low level of P1 promoter in both constructs. So this low level of hunchback is sufficient for early hunchback function. To answer this, we deleted the 120 base pair core regions of P2 promoter using the CRISPR technique. We firstly replaced it by a DS-read report and then flipped it out. The mutant cuticle lacks T2 and T3 segments compared to the wild-type ones, so indicating that P2 promoter is absolutely critical for the hunchback activation, and the P1 promoter cannot substitute for it. And this also further confirms that the P1 promoter could not be activated by Bicoid-dependent enhancers, and no matter P2 is present or not. So back to the question, why is that? Is there something within the promoter sequence that controlling the specificity, or is there something in the locus that blocking the enhancer-promoter interactions? To distinguish between the two possible mechanisms, we replaced the core promoter region of P1 with P2s. So if now the new promoter, if this new chimera promoter can be activated, then it suggests that the promoter sequence is controlling this specific enhancer-promoter interaction. If this promoter, the new promoter cannot be activated, then it suggests that the location of the promoter is more important. Okay, so here shows the results. After replacing the P1's core promoter region, it's also 120 pair bases. Okay, so the level of lacZ, the N-half lacZ increases about more than 5-fold compared to that in the wild-type construct, suggesting that there is something within the core promoter region that's controlling this specificity. But also, as you can see, although the expression level increased a lot, but it's not as high as the original P1 promoter, as you can show in here. So it suggests that the location of the promoter at the locus is also important. But here we only focus on the sequence part. So to identify the elements in the promoter that are controlling this process, we've compared the sequence of the two promoters, and we found that the P2 promoter has two overlapping initiator motifs and two Tata boxes. And this feature is conserved across 12 Drosophila species. And so we're guessing this is the main feature that making the two promoters different. So right now we're in the process of testing if adding the double Tata and the initiate to the P1 promoter can convert it into a Bicoid-dependent promoter. Okay, to summarize, first, we found that the P2 promoter is critical for hunchback activation and P1 cannot substitute for it. And the second, with some data not shown, we found that the distal enhancer specifically interact with P2 promoter, and this contributes to the robustness of hunchback expression. And also, the 120 base pair sequences of the P1 and the P2 promoters control the specific interactions with Bicoid-dependent enhancers. And the double tata and the initiator motifs in the P2 promoter might be the molecular basis that enables the Bicoid-dependent activations. And thanks for all the members in small lab and also Thomas Grab lab, especially Sean, who kindly shared me with all the single molecule fish stuff. And glad to take questions.
Hi, I was wondering, you said you put two P2 promoters to test for sequence. Did you put two P1 promoters to test whether location is different, whether the location plays a role?
Sorry, I can't hear you clearly.
Oh, sorry. Did you put two P1 promoters in instead of two P2 promoters? If location was important, you would expect to see an increase of the N-terminal?
Yes, that's something I didn't show, but if I put the two P1s, so if I understand you correctly, if I put the P1 after the proximal enhancer, it can be activated to a level similar to the P2 promoter. Is that what you asked?
So if instead of in your regular construct, instead just replacing the P2 promoter with the P1 promoter and having the other P1 promoter intact, if location, specifically location, plays a role, you would still expect a slight increase. You said that sequence accounts for some of the specificity of the P2 promoter, but you don't rescue entirely the expression, right? So if also location is important, then you would expect to see some effect if both of your promoters are P1 promoters.
Yeah, you're right. So if you replace the P2 promoter with P1, it could be activated. As I didn't say that because the proximal enhancer is very sensitive to the distance between promoters. Because of P2, so if you replace the P2 with P1, then it's right next to the proximal enhancer, and it could be activated by it.
Two quick technical questions. So how big is the distance between P1 and P2 promoters?
It's about 3 kilobases.
3 kilobases. One more thing, have you tried injecting this at a different places, chromosomal locations? You did ATTP injections, I suppose?
No, not yet. That's something I can try.
Hi, nice talk. So in my lab, we've done some open chromatin profiling in early embryos, and if I recall correctly, at blastoderm stage embryos, the P2 promoter is open and accessible, whereas the P1 promoter is not open. So I'm wondering if part of this is regulating accessibility of the P1 promoter, and I guess you could test that by adding maybe some Zelda sites to the P1 promoter to see if that could allow it to be activated at earlier stages.
I'm not sure about the time resolution of your chromatin accessibilities, but my experiment is done at the early cycle 14, but from middle cycle 14, the P1 promoter could be activated by a stripe enhancer. So I don't think that locus is closed.
Okay, thank you.
Thank you.